Thank you. Okay, thank you very much, first of all, to the organizers for this really amazing workshop. I've enjoyed very much hearing all the talks, and the level of the discussion has been really great during the talks as well. So I'm going to talk about cell polarization and motility. Before I do that, let me just emphasize that this work was done by many, many people who I had the fortune to be associated with. These are some of them, and so Yoichiro Mori, who's actually in the audience, was a former postdoc. Now he's an associate professor in University of Minnesota. Sasha Zhilkin and Adriana Dawes were former PhD students, as were Corey Simon and May Ann Mata, and the rest of them, Ben Vanderlee, Bill Holmes and Stan Murray, are former postdocs, and Veronica is a partner with Stan Murray. Now, before I actually talk about cell motility and polarization, I want to spend just a little bit of time introducing you to GTPases, to Rho GTPases. And I'm going to do this in the context of a completely separate project that's not really about cell polarization, but I think it very well illustrates what these Rho GTPases are capable of and how we can visualize them. So this is actually work by Bill Bement from Wisconsin, and what he does is he takes frog eggs and makes tiny little wounds. These are now single cell wounds on the surface of that egg. And what he sees happening on that surface is the wound closes, and associated with that closure are bands of Rho GTPases. So he's able to visualize two of these proteins that sit in the membrane, and these are Rho, shown in green, and CDC42, shown in red. Now, it turns out that CDC42 is recruiting actin filaments, assembling or aiding to the nucleation of actin filaments, and Rho is what's causing myosin activation to contract this wound in a purse string-like manner. And so this is basically a set of interactions that through some experiments he was able to provide evidence for. So these two types of proteins, CDC42 and Rho, interact with one another through some kind of mutual inhibition, and there's some other protein that activates Rho, etc. And I've already mentioned that one of these, they both help to repair the wound either by recruiting actin or activating myosin contraction. So very briefly, Corey Simon, then a PhD student with me, hooked together a bunch of different models to try and understand how these beautiful bands can form and sort of self-segregate around the closing wound. He looked at a variety of models. These were spatial around, radial coordinates around the wound. I'm not going to go into great detail here because I want to move on to the other stories, but he was able to show that a certain model, model 3, the one that has additional feedbacks, can account for the data. Namely, if you look at some initial distribution at some time after wounding, as it's seen in the experiment, then you see that gradually these zones, green here is the Rho zone, red is the CDC42 zone, this is distance away from the wound center, and they will segregate and form these two bands. Okay, so essentially that was our first work with Bill Bement. Now Bement also does all kinds of knockouts. He's able to suppress Rho, suppress ABR, so forth. And so, of course, we challenged the model to see if we could account for these knockouts, and it turned out that it did pretty well. And again, I'm not going to spend a lot of detail here. I just want to mention this because it was a fun experimental collaboration. And also, there was one experiment done just for us, namely Bement made 2 wounds at various distances from one another, and the model was very nicely able to account for the type of patterns that you get depending on how close or how far away those 2 wounds were. Okay, so
sorry, just for the sake of curiosity,
yes.
How does he puncture those cells?
Laser.
Oh, it's
He
laser
uses a laser.
surgery.
Yes, exactly.
Leah, just one question.
Yes.
Are the cells viable once the holes have healed?
Yes.
Yeah.
Oh,
Okay.
absolutely. Yep. They're able to.
Nothing goes out of the hole? There's no material?
I'm sorry?
Isn't there material going out of the hole once?
Not really. No. So somehow it's able to keep itself together. I mean, if the wound was very big, I imagine the whole thing would blow apart. But these are rather small wounds, and it's a beautiful experimental system. I encourage people to have a look at some of the papers by Bement because it's really quite lovely. Okay, I want to go on to the main topic of the day, which is cell polarization. I have a feeling that this may not be working anymore. I think the battery may be dead here. Let's see. Yes, the battery is dead. So I'll just do this. Right, I'll do this manually with a stick. The low-tech method always works. Okay, so now we've heard a great deal about actin, cell motility, cell polarization, so that's wonderful. I can breeze through the first few slides. Let me just remind you that cell polarization is in general this symmetry-breaking event where a cell that's initially disc-shaped or somehow symmetric or nearly symmetric undergoes this transition to a polarized state. And we've seen many examples of cell motility. I don't have movies because I didn't want this to crash, but Alex certainly showed us beautiful movies, and so did many other speakers here. And so by now you know that cell motility involves, to a great extent, protrusion of the front edge of a cell, as well as retraction of the rear, and that this is organized by orchestrating actin and myosin and many other things in the cell. Okay, great. So we've also seen that the front edge of a cell, for example, in a keratocyte, has this meshwork of actin. I'm on purpose using meshwork and not network. That was a point of confusion. And the rear has myosin that helps to contract and pull up that rear. We've also seen this picture many times in I think, at least 2 talks. And so here we have the actin cycle where, actually you see this better, I think. So this is the front edge, and you've got actin being assembled, branched, nucleated, and in the back it depolymerizes. Now, how is this orchestrated and how is this regulated? That's really the topic that I'm going to talk about. And so if you look at protein networks in the sense of graph theoretic networks that regulate actin, if you look this up online, you see pictures and images that look much like this. Now, to orient you, here again is the cell edge. These are receptors up here. They sense a variety of signals, funnel those signals down somehow through all of this molecular circuitry, and eventually impinge on actin polymerization as well as myosin contraction. So one question anyone might have when you look at this is, well, how do we understand this? Even given the fact that the arrows don't all mean the same thing, right? Some arrows mean this binds to the other. Some arrows mean this gets transformed to something else. So it's quite confusing. Now, just to orient you a little bit, there's an important layer that we'll spend quite a bit of time on, and I've already alluded to, and these are called the Rho GTPases. I've already introduced you to CDC42 and Rho, and so in a way you've already seen that they somehow can form patterns on the surface of a cell. And it turns out that these are important master regulators of many, many cell functions, including cell motility including the actin cytoskeleton and myosin, actomyosin assembly. So let me tell you a little bit more about these. Again, two of these are dominantly associated with actin nucleation via proteins on the membrane that activate, this should be ARP23, which then causes local actin branching. The other guy, Rho, does a variety of things, including activating myosin contraction and also formins. We heard a little bit about formins yesterday. I'm not going to deal much with that aspect of oral function in my talk. Now, in motile cells, it turns out that these proteins form remarkable patterns on a time scale of about seconds. These are patterns in the cell, right, on its surface. And here you see, for instance, RAC and CDC42, which commonly are associated with the front of the cell, where actin is being assembled, and Rho, which quite often is found in the back of a cell, not always, but quite often, associated with rear retraction. Now, the other beautiful thing is that we also have experimental work that shows that these are dynamic patterns. In fact, you see wave-like dynamics. And some of these GTPases, like RAC, this is work from the lab of Orion Weiner. So the beauty is that we can visualize these proteins. We can see how they respond in time. Now the question is, how are we going to go about trying to decipher these signaling networks? Well, the way we went about it is sort of to dissect it into a bunch of layers and kind of try and isolate each layer at a time, study it, and then see if we can hook them up together. I'm going to later mention an additional layer. Let's leave that for the moment. We'll come back to it. But this, again, the PI3 kinase is also a protein that's known to be upregulated at the front of a cell, in particular in Dicty. Okay, so now let me orient you to what is it that I would like to focus attention on. The first part of the talk is going to really focus on chemical polarization. So this will be quite distinct from what Alex told us about yesterday, which was really polarization due to some kind of mechanical push or de-adhesion or something like that. I'm going to talk mainly about how the Rho GTPases polarize inside the cell, and then later I'll talk a little bit about how that in turn gives rise to the cell changing its shape and actually moving via the actin engine. So first, a little bit of history. If you look back in the 2000s, which is a mere 15 years ago, so short on the time scale of mathematics, but eons on the time scale of biology, then even back then there were some ideas about how these various GTPases talk to one another. And we looked at one in particular that had mutual inhibition, and that looked intriguing and interesting because we know that those type of systems have the property called bistability. So we were hoping that somehow this would give rise to the type of polarization that we were trying to explain. Now this is work that was due to Sasha Zhilkin and Stan Murray. We looked at these sort of interactions, and we looked at a sort of one-dimensional cell transect. And now let me make a point here which I think is quite important and potentially confusing. And that is, there are some works, for example, this is just one slide from the PhD of Teng-Hu Wu, who just finished his doctorate with Jimmy Wu. Jimmy's here this week. What they do is they actually model the chemistry on the perimeter of a domain. In this particular case, the cell was basically flowing into a slit and became activated as a result. So quite commonly in the literature, you see chemistry modeled on the perimeter, where people say this is the cell membrane or the edge of the cell, and this is the cell interior. But that's not what I'm doing. That's not what my group has been doing. Rather, we're looking at 1D, which is a diameter of a cell, right? So you'll see a variety of geometries in what I talk about today, and they'll either be 1D, which means I'm looking front to back, right? So one end of the domain is the rear and one end is the front. And of course, I'm getting rid of the cell body. I'm assuming the cell has uniform thickness as though it's a fragment of a, let's say, keratocyte. And sometimes I'll revert to talking about a 2D version of the model. Again, I'm getting rid of the cell body. I'm looking at a thin sheet, but that sheet includes the top surface, the membrane, the interior, and the bottom surface. So that's just a point to keep in mind. Okay, so back to the model that we initially set up. We basically put these interactions into functional forms using sort of typical inhibition and autocatalysis functions. 3 PDEs with diffusion, activation rates, inactivation rates of each of these 3 GTPases in 1 dimension. And we simulated this. And we were very disappointed because it would give us robust polarization. So what I'm showing here is typically what you'd see in a chymograph. This is space and this is time. And so here you're seeing the diameter of the cell with the activity profile of CDC42. And what we see here is that you get some wave of polarization. It sweeps all the way through the cell. The entire cell gets active CDC42, and that's no good, right? Because at the end, we don't really have a front. We don't have a back. We have a uniform activity level. So in hindsight, that seemed to be fairly clear why it happens. It happens basically because when you hook together bistable kinetics with diffusion, you tend to get traveling waves. But of course, we didn't quite think of it to start off with. Okay, so back to the drawing board, and here's where the biology actually turned out to be very important. So we went back to details that we had left out. What are these details? So it turns out that GTPases come in, well, they have several states. This is a simplification. When they're active, they're bound to the plasma membrane. They can be inactivated and they'll come off the membrane, and there's back and forth cycling between these 2 states. There are proteins that mediate these activations and inactivations. They're called GEFs and GAPs. There are also proteins that mediate the GTPase coming off the membrane. I'm leaving all that off of this cartoon. Even this in itself, what I'm showing here, is really quite important, as you'll see. Now, when we included these inactive forms, so now we've got 6 PDEs, and again, let me explain the geometry because I think this is potentially confusing. I want to make sure that it's clear. So what are we doing? Well, let's look at a magnified portion of the cell. The active proteins, they're actually sitting in the membrane, and the inactive ones are inside the so-called fluid, the cytosol. But we're not going to model this as 3 compartments or even 2. We're going to say they're all in some homogenized domain. We're kind of thinking of this as homogenized vertically, but there's one specific property that's different between the active and inactive ones. The active ones are diffusing in the plane of the membrane. That means that they diffuse rather slowly compared with the ones that are in the cytosol. That's the only difference. So again, it's a caricature, but it captures important details. All right, so now we have 6 PDEs. It's still in 1 dimension. These are the active forms. These are the inactive forms. We've got conservation. So, for instance, this term, which is the rate of activation and inactivation, shows up here as the with a minus sign, right? So the total amount of CDC42 is conserved, and so is the total amount of RAC etc.
Question?
Yes, please.
But is that true point-wise rather than just in an integral sense?
It's true in the integral sense. In the integral sense, yeah. So the total amount in the domain, I should say, is conserved. Yeah. Notice that the rates of diffusion here are not the same. These guys are in the membrane. This is in the, well, this is some intermediate between membrane and cytosol, but this will be an important aspect.
And the second equation is that Laplace of CR, product of two functions.
This is a typo. This should be just R. I apologize. It's an old slide and I didn't notice. So these are just plain old diffusion. There's no cross diffusion. Simple reaction diffusion. Very straightforward. Okay, lo and behold, the wave stops in the middle. So all of a sudden, just by including these inactive GTPases, which supposedly do nothing because they're inactive, right? Then we get that the wave will enter the domain and it'll freeze, it'll stop in the middle, and so as a result we get net polarization with some highly active level of something in the front, low level in the back. Yes?
Quick question. Whenever you refer to that particular behavior, you refer to some particular parameter set or regime? Like
this is robust for a whole range of parameters.
Okay.
Now, not for every parameter. I'll come back to this, actually. So this is not for one cherry-picked value of the parameters. And that's really a very interesting and great question. Yes? Yes?
Can you say something to the concentration of the GTPases?
You can. So I won't do that here. We can discuss that offline. Please keep in mind that we don't really know the exact functional kinetic terms to use because nobody's done the exact biochemical work to tell us precisely how the GEFs and GAFs are. We don't have enough detail to quantify everything, but what we do know are rates of turnover. We know steady state levels. We can quantify quite a lot of we certainly know rates of diffusion. So there's a lot we know, but there's also a lot we don't know. In other words, the model is a caricature. Please view this as kind of a plausible model, but it's not necessarily biologically exact. Yes?
Have you got an intuitive explanation for what stabilizes the wave front?
That's coming.
Oh, right. Okay.
Yeah, so hold on to your hat. That's coming. All right, so first, just a couple of general points, since Hans said that we should, I don't know, make points and raise questions for the audience. So first of all, for the young people, if your model doesn't work, actually that could be a very good thing, right? Because it tells you something is missing. And so it actually informs you that, well, it's not as simple as the biologist might think or whatever. Okay, so I think I've mentioned this already. We could go on. Right. So, so far, remember, we're looking at just one layer of signaling, which is the rho GTPase layer. And the question is, we think we might understand something about that layer. Can we somehow connect it to some of the other layers? So that's what I'll briefly mention next, the fact that rho GTPases actually talk to the actin cytoskeleton. Now let's see what happens when you hook both of these up together. And you hook them up together by assuming that these two are responsible for actin assembly, and rho, again, is responsible for rear retraction through myosin contraction. Now, let me mention that, again, for the benefit of the young people, when we did this, we always go through a bunch of preparatory steps before we look at cell shapes. For example, we first look at well-mixed versions of the equations, and then we look at one spatial dimension, and then only then we look at more complicated geometries and so on. Because you can save yourself a lot of time by realizing things don't work well here and tweaking and refining them before you go on. Right, so to put this model into 2D and include the actin layer, this is really work by Stan Murray. It was published in the bulletin. I can give you the reference later. and it's based on a so-called cellular pots model. Now here I won't have enough time to do justice to this sort of model, but let me mention briefly that it is a kind of stochastic agent-based model. We've got the world's expert sitting in the front row here, and so you can corner him later to find out more details. But basically what's happening is, think of a small piece of the cell. At each step, what you're going to ask is whether a little piece of that cell tends to protrude or retract, and there'll be some kind of a balance that's tested using a Hamiltonian that takes into account whether the cell volume, or in this case area, is too big or too small, which would make you tend to protrude or retract. I'll be right with you. Whether you've got local myosin, which tends to make you want to contract, or whether you've got pushing actin ends that tend to propel you forward. Yes?
Quick question. Is there a
Yes.
particular advantage of using hexagonal grids rather than...
Yes. In this particular case, we used a hexagonal grid because we wanted to have actin filaments that branch. We wanted to incorporate ARP23 branching. Now that happens at 70-degree angle, but 60 degrees is close enough. And so we had like 6 different orientations of actin filaments, and they gave rise to daughters of plus and minus 60 degrees. Does that help? So a hexagonal grid was useful for this.
Are you measuring the cell volume? Is it the whole cell or the individual computational cell?
It's this area. So the cell volume is actually an area in 2D. It's all 2D here, right?
Okay.
And so there is a, well, we can ask Dr. Glaser later, the Hamiltonian has terms that penalize cell area getting too big or too small, perimeter getting too large or too small, etc. Okay. Where was I? Yes, so there's a lot of equations. I'm not going to spend time on this except to say, just to orient you, there are the GTPases, the inactive forms of them. There's ARP23. Notice it's being upregulated, being activated, I should say, by CDC and RAC. It goes into actin branches, etc. The actin filaments, well, there's branching, creation of new barbed ends, etc. I'm not going to go through all the details, but basically all that was put in. And so it turns out that just with that, you're able to start off with a symmetric cell, get it to polarize rather quickly, and get a high level of the CDC42 and RAC in the front, a low level of those in the back, and you even get the cell to respond to changes in the signal. Now what is the signal? The signal is basically a rate of activation of, let's say, CDC42, a transient, small pulse of a gradient of CDC42 activation across the cell for a short period of time, remove it, but the cell polarizes, and so it can do something based on that. Right. Okay, so let me just mention that in more recent work with Stan and Veronica, we were able to include the additional layer of phosphoinositides. All of a sudden the diagram becomes rather complex looking, but really this layer is doing most of the heavy lifting. And this layer, notice PI3K and P10, these are known to go to the front and back of Dicty cells, for example. This layer turns out to fine-tune the responses in the following sense. So let me just briefly show a movie. So notice that there's feedback here between PIP3, which is a membrane lipid, it's a phosphoinositide, and RAC, and there's sort of a positive feedback loop. But the strength of that feedback has to be somewhere adjusted. It cannot be all the way up to very high level, because then the cell tends to sort of get stuck. And it cannot be too low, because then the cell doesn't know what to do when it encounters a barrier, and it tends to sort of squash itself against the wall. But as you saw in the movie, if that feedback is about right, here f is 0.4 on some normalized scale from 0 to 1, then the cell is able to orient itself when it bounces into a wall and go up or down. I want to make another point, which we discuss in the same publication, that I think one would miss if one was doing computations on the edge of a cell, as I mentioned earlier. And that is that the cell shape itself turns out to have a feedback onto the rho GTPA's dynamics. And this is purely a kind of motion by curvature effect, which mathematicians know about, but I think one would tend to miss if you didn't know to look for it. So what we're showing here are two time sequences. In this one, the computational cell was treated with electrunculin, which means it cannot change its shape. There's no actin cytoskeleton. And all that's happening is it's initially polarized in one direction, and then a stimulus in the opposite direction gets it to repolarize. Now, you see it takes it some time, and eventually it's halfway there. But notice what happens to the cell that is got the shape-changing ability. This happens much faster. So over here, notice the cell is almost completely repolarized. These were taken at the same time steps, same amount of time. Now, why is it happening? Well, briefly, it's happening because at the boundary of the cell, the contours of the equal concentration have to meet the boundary at 90 degrees. That's something that you can show from the no-flux boundary conditions. And so the fact that the shape changes means that these little pieces of the chemical contours have to keep up with the edge. And this, in turn, has the property that places where high curvature develops have a much faster dynamics. So this is something kind of interesting, because it really shows that understanding the mathematical underpinning and this motion by curvature can give us some insight into this. I think this may also say something biologically, but I won't swear by it. Okay. Break for a few general points. First of all, as I mentioned, it's a good idea to start with simple geometry, but keep in mind that changing the geometry can produce new effects. And I already mentioned that this is true in what we've just looked at, and motion by curvature. Okay, now a little bit of head scratching, and that is, do we really believe this model? And the answer is obviously no, right? I mean, all models are toy models, and you use them, you get something out of them, and you throw them away. For example, we might have the wrong interactions. We might not necessarily have mutual inhibition between CDC42 and Rho. We might have the wrong parameters. So it's really important to try and extract what are general principles, what are the key underlying mechanisms, and how can we understand what they do biologically? Under what conditions will such a regulatory module work? Is it just a chance, or can we say something more general? And so that's where it's nice to use mathematical techniques and go back to a simpler model, a cartoon model, as Hans has nicely put it. So this is the cartoon model. Now we'll go back to forget about 3 GTPases. Let's just look at one in an active and an inactive form. let's look at what we could get out of just one GTPase. And this, I should say, is predominantly work of Sasha Zhilkin and Yoichiro Mori, who's, as I mentioned, is here this week. So what did we assume? Well, first of all, we know that diffusion of proteins in the membrane is far slower than proteins in the cytosol by a factor of 100 to 1,000. We've also assumed some kind of positive feedback, otherwise you don't get anything interesting whatsoever. You must have some nonlinear kinetics to get the thing to stop. And so we looked at a model where you've got an active form, this is its concentration, an inactive form, there it's its concentration. They diffuse in one dimension with different rates, and they're being exchanged. And the function we used here was essentially something that has this cubic type of shape as a function of u, and it also depends on v. So the nice thing here is that because this is simple, well, relatively simple, only 2 PDEs, you can actually do some hard analysis on it, and that's what Yoichiro did. You can rescale the problem and take advantage of the fact that there's a slow parameter. This is something applied mathematicians love to find, a slow parameter. Also note that there is conservation. This plays an important role in the behavior. So here I'm not going to describe all of the great details that went into it. Maybe later this time we can go more into that. But it turns out that by doing that analysis, you're able to find that the wave that we saw earlier is created through this positive feedback. It propagates by the nonlinear reaction diffusion interactions, but it's halted essentially by depletion, using up the inactive protein. And that's why conservation is important. So you can also find out when wave peening won't happen. So under what conditions would you lose it? You can do this by looking at some bifurcation diagram. This is just one of many. And you see that this parameter epsilon actually influences whether you're in the zone, in the parameter plane, both the total amount and epsilon, whether you'll have wave pinning or not. And so moving this way on the diagram is equivalent to either increasing the diffusion of the active protein, increasing the domain size, or reducing the reaction rate. In other words, you're going to lose the ability to polarize if any of these things is true. If your cell is too small, if the diffusion of the active protein is too fast, or if the activation and inactivation rates are too slow. So we learned something biologically. In particular, we learned that cells that are too small may not be able to polarize using this mechanism. Now, I should mention that, again, Yoichiro did some amazing work on the detailed bifurcation behavior of this system. This was published in SciAm. I'm not going to talk about that. There's a lot there for the mathematicians to look at. But the discouraging thing about that one example, going back to this slide, is that it takes a lot of work to understand how PDEs behave. So doing full bifurcation of a pair of PDEs even, like this is the absolute rock bottom, simplest model, well, that's a lot of work. And when you change the PDEs ever so slightly, by changing one kinetic term, your whole work, you can throw it out the window and start again, right? So we realized that we need a shortcut for analyzing at least approximate behavior of these sort of systems. In particular, these kind of slow, fast reaction diffusion models, slow, fast because of this fact that the active proteins diffuse slowly. The inactive ones diffuse quickly. And we needed some kind of shortcut that's scalable to larger systems. And that's, in fact, what I'd like to discuss next. Now, this is, we call it local perturbation analysis. It really was invented by Stan, who calls himself a biologist, but he's also a great mathematician. And it appeared in the appendix of a PhD by Veronica, but that's sort of where it lay hidden until Bill, who I mentioned was a postdoc at the time, tried it out on a couple of systems, and he came back and he told me, I am shocked how well this works. And so then we started to use it a great deal. Now, this also has been sort of published in a user-friendly, with user guides recently. If anyone's interested, I'll give you the reference later. So essentially, what is it? So remember, we've got, for example, this pair of PDEs with very different rates of diffusion. What we're going to ask is a very simple question. Suppose we have the uniform steady state of that set of PDEs, and we superimpose a arbitrarily tall but very narrow pulse. So think of this as taking the cell and kind of poking it in one place. And the question is, what's going to happen? Is this pulse, is this local perturbation going to grow into some kind of interesting response, or will it just fizzle and decay away back to nothing? In other words, will something interesting happen or not? So to answer just that question, the behavior of this pulse, it turns out that you can approximate that by looking at ODEs for the height of the pulse and the levels of the background U and V. In other words, one local and two global variables. Now, why is that useful? It's useful because when you reduce a PDE model to ODEs, you can use bifurcation software, which is ready-made. You can use MatCont or XPP or whatever. And so you can come up with a diagram that summarizes the behavior of these so-called local perturbation ODEs. So let me orient you what we're seeing here. So here, this particular example is for the same wave pinning two PDEs with those kind of cubic-looking reaction kinetics. And what's plotted here is, this is some parameter. In this case, it's the basal activation rate, and plotted against the height of that local peak. Now, the black curve, if it was there by itself, it would signify the behavior of just the well-mixed system. So if you look at only the global variables, you would get a very boring black curve, which means that you have one stable steady state and nothing else interesting. However, when you hook it up to this ODE that describes the height of the peak and how it depends on the other variables, you get this additional set of branches, and they're telling you something. So what are they telling you? They're telling you that if the height of the peak is too small, and you don't, for a given parameter value, you don't breach this threshold, you're going to just, that peak will decay back down, and you'll get back to the homogeneous steady state. However, if that peak height, the perturbation, is above that threshold, then something else happens. Some more interesting behavior happens. It grows, and then we can no longer say what happens. We've got to look at the full PDEs. Now here's another domain. In fact, you can separate the behavior on this diagram into several domains, two of which, there's another thin one here, have this sort of threshold behavior. And then in the other domain, no matter how small that peak is, it will always give rise to some pattern. And finally, there's a domain in which, a parameter region, in which you always get back your homogeneous steady state. Nothing interesting happens. Yes?
How does that evolution equation for the local dynamics differ from the equations for the homogeneous system? I see the same F.
It's UL here interacting with VG.
So the VG is the
VG is the background. So essentially the pulse
So you
is,
freeze the V at the steady state, look at the resulting dynamics.
well, the V is not quite frozen because, I mean, it also satisfies this, right? But it's basically saying that this little pulse is feeding off the background, and interacting with it, but it doesn't affect the background. That's basically what it's saying. This is not rigorous rocket science math, but it turns out to be a very handy approximation, and you'll see in a minute why. Okay, so first of all, just quickly, we can compare this sort of signature of the model, of the wave pinning model, to some of the other models that are popular in the literature. So this is Otsuji, and here's Meinhardt, and you see that this one has a particularly nice threshold domain, as well as a resting no response domain. And by the way, these are the full PDE simulations. Again,
But your ODs are spatially independent.
correct. The ODEs are just for the purpose of producing these sort of handy, call them bifurcation
The
diagrams.
numerical
The
solutions
PDEs are
Yeah.
are what produce these space-time plots. The full, so again, the LPA, the analysis, only helps you to figure out what interesting parameters to use. Then you have to go to your full PDE solver, but you get more insight, right? You have some idea of how to tweak your parameters.
And your red curve, is that the solution of your...
The red curve here is the branch in the bifurcation diagram that's basically due to this local pulse. Okay. Based on what I've just told you, I want to just interject, and I need to speed up a little bit because I'm going to...
There is 1 hour.
Yeah, I know there's extra time, but I don't want to go for an hour and a half. That would be like...
Okay.
Okay. So let me show you how that same LPA was useful in understanding an actin wave model. So what we did was hooked up, again, the same wave pinning 2 PDEs to one additional variable, F actin, and we made the assumption that, let's say, CDC or RAC activates the assembly of actin. Actin disassembles, but actin also inactivates RAC or CDC42 or whatever it is, this nucleation promoting factor. And we asked, how would this behave? So on this side, notice it's just 2 PDEs. All we've done is we've hooked one more PDE to it. In fact, well, PDE without any diffusion or anything. And this, the activation rate of the GTPAs depends now on F-actin. I should say the inactivation rate depends on it. In other words, this arrow right here for inactivation shows up in one of the terms that's sitting in here. And actin is being produced at a rate dependent on the nucleation factor. All right, so notice that the LPA diagrams are kind of informative because they show you exactly what new features come out. Namely, you get a couple of new hop bifurcations that you didn't have before. And further, a comparison, this basically always, within the wave pinning regime, polarizes. But here you can get much more interesting kind of dynamic waves. So this is a far simpler actin wave model than the one that we heard about from Hans. A variety of different kinds of wave-like behavior. There's no stochasticity here. It's just different parameter settings. And we're able to understand it, again, using the, and this time, 2-parameter bifurcation plot based on the LPA mechanism. This fishtail-looking thing is the tracking the hop bifurcation. And the different types of dynamics were found in different regions of the plane here. Again, I emphasize that to actually see the space-time dynamics, you have to solve the PDEs. You cannot deduce any of that directly from the LPA. Okay, I'm almost at the end, but I didn't want to stop before I tell you a little bit about how some of the cell polarization theories have been connected to experiments. So I've been really lucky to be associated with Andrei Levchenko and his group, in particular Benjamin Lin, did a PhD with Andre, and they did something really quite interesting. So what they did is they used microfluidic devices, and they put HeLa cells in those chambers. And those HeLa cells had specific constructs in them whereby they could directly control RAC, the activity level of RAC, without its usual upstream regulators, just using a small molecule, rapamycin. Now, the details of how they did that, you'll have to read in their paper, but all I can tell you is that essentially you're in the driver's seat controlling what RAC is doing. So what they did is they can very accurately control the gradients of this small molecule rapamycin. So here's a cell inside one of these microfluidic devices, and you see the red dye here is showing you that there is a gradient of something flowing across. And you also see that as a function of time, that cell has polarized and started to move in this one-dimensional chamber. So the 1D is really a nice reduction because the cells are constrained, they can only move up or down. Right, and as a result, you also see that RAC becomes graded inside the cell, right? So here, very high activity level of RAC at the front of the cell. So what they did is they studied the polarization responses to different initial cell states as a function of the strength, the duration, the timing of the gradients, and they characterize some of those responses. So these are chymographs that show that, for example, this cell, this is time, this is the length of the cell, that it took it a little bit of time and eventually it did some kind of response. And there was some quantification. I should mention, though, that we, in parallel, we hooked up several type of models and examined which of them might be suitable for understanding the system. We had to revise the model that I've described earlier because it turned out, something we had not realized before, that when you take that model I talked about earlier and you stimulate RAC, the cell polarizes in the wrong direction. So we were able to reject our old model, and we examined a variety of others, and for reasons which I'll show you in a minute, we settled on this one as being capable of explaining the data. And at that time we thought that CDC42 was also important, so we added that as well. Excuse me, let me just take a quick drink here. Now let me show you what sort of quantification was used to weed out some of the models. So for example, the effect of the gradient the effect of the gradient on the initial response time. So the experiments show that as you increase the gradient, this is the level of the gradient here, this is the time it takes a cell to respond, the higher the gradient, the faster cells respond. Same thing with the model. Again, here the gradient of the rapamycin is represented by a slope S1 and by a kind of baseline height S0. There was also an effect of the stimulus strength on a secondary response. So you see that if rapamycin was only introduced for a very short time and washed out, the cell basically does something but is very gradual, as distinct from if you have a large enough amount of rapamycin, or rather long enough duration, you get this secondary response, which is kind of a threshold based response. And the model was able to account for that as well. And finally, looking at the effect of knockout of PI3K, which presumably breaks this feedback loop that we think might be important. This was done by the inhibitor, this PI3K inhibitor, and you find that cells take longer to polarize depending on what state they are in, and the model does the same. A final couple of comments before I stop. I'm almost out of time. It's intriguing that the LPA diagram for this little GTPase circuit, which I've shown you, has many features that are quite similar to the LPA diagram for this much more complicated model that we certainly could not analyze using PDE bifurcations. Namely, there's a region over here where you've got sort of a threshold response that's very similar to this region here, and so on and so forth. And one other comment I wanted to make, and that is the fact that it looks like there are all kinds of universal themes that we see popping up in many distinct cell types. So this is a diagram that was in the talk that Hans gave for dictyostelium. And we see that there are these PI3K and PIP-P10 which affect the phosphoinositides, and we see that there is RAC, we see that there are feedbacks between them, and we see similar things in, for example, the HeLa cells that we've been looking at. More recently, we're now less focused on CDC42. We're really paying more attention to mutual inhibition between RAC and Rho. I won't have time to describe any of those more current projects. You can catch me later for that. But here are a few comments to kind of put in some caveats and put it together. So first of all, we've left out a lot of really important stuff, which is clear. We haven't put in a very important link between the mechanics, like membrane tension and adhesion and all of that, and the signaling. That is known to play a huge role. So in the future, that would be really important to put in. I hope I've persuaded you that it's kind of fruitful to go between simple models where you can play with them mathematically or get some brilliant mathematician to analyze them, and the ones that are more detailed that you can actually tweak parameters that have some biological connection, and also that it's really useful to go from 1 to 2D and vice versa to explore these models. Also, I think that there's this general idea that I'm pretty convinced, well, okay, I hope some of you are too, that math really can sometimes uncover basic principles, underlying principles, where intuition or simple verbal arguments fail to do so. And we've already talked about this universality principle. So with that, I'll just thank you, and I'm open for questions. Yes.
So when you introduced wave spinning model, it sounded like your motivation for choosing cubic nonlinearity was that you wanted to see something interesting. It's a little bit unsatisfactory to me, so my question is, can you actually take a larger model, one of the ones that you showed later, and somehow cross-grain or reduce them to simpler form, but with a more complex reaction term? And would you come up with the cubic nonlinearity or something like that?
Well,
possible?
Right. So in the 2 PDE variant that I talked about, Yoichiro actually showed the necessary conditions for wave pinning. And I think I've got these somewhere. Let me just find that. And it turns out that you need something that has a cubic shape, or else you're not going to get anything interesting at all. So
I can believe that, but it doesn't come from biology, right? So, but it might come.
Well, it does tell you something about the biology. It tells you that you have to have some kind of, first of all, nonlinear interactions for sure, and you have to have some kind of either positive feedback or mutually inhibitory feedback to get something like this.
Yes, I understand, but still, like particular form, why not 5th or 5th order, 10th order, or whatever? Like
Oh, yeah.
You can get
Oh,
all kind of
oh,
interesting
yeah. Oh,
stuff when
excuse
you
me.
make-
Oh, it doesn't have to be cubic at all. As long as it has a shape like
But
this.
could this emerge from the network, from the protein network that you consider by
That's a good
reduction?
question. That's a good question, and I'm not sure, but I think that's a very fair question. It would be nice to, if we could show that.
Well, in that same vein, did you make a more detailed model for the last step where you had the CDC and the row rack and the PI3K, etc.?
Yeah.
That was a detailed molecular model, or
Well, we don't know the details. If we knew the details, it would be very nice. But what I showed way back here, I mean, so this is as detailed as we got, basically.
but I mean at the point of writing down the differential equations. but-
Yeah. Oh, yeah. Oh, we have differential equations for all of this stuff. Yeah. This is in this paper here. I'm happy to send you more. Yeah, it's easy to. Yes.
On your first part of your conclusion, you said you didn't have any mechanics. So could you go into a little bit more about what the cellular POTS model, how that's creating the movement?
Right. Okay. So I don't have more detailed slides than what I showed, but let's go back to this slide for a minute, which is a cartoon of the cellular POTS model. So in the cellular POTS model, you take, for example, a two-dimensional grid, and you carve it up into domains, part of which could be the domain corresponding to the interior of the cell, or the cell, let's say, not the interior of the cell. So pretend that this is inside a grid, and here is my cell domain. Here's a blown up view that shows actually the grid. The grid happens to be hexagonal in this particular realization, but usually it's rectangular. Okay? Now, at every so-called Monte Carlo step, what happens is you go through randomly through a variety of these grid points along the cell perimeter, and you ask a simple question. You flip a coin and you ask a question: Is it likely that this piece will protrude outwards or will be retracted inwards? In other words, will this change state from being part of the cell to being part of empty space or not?
So at each step, volume's not conserved. You might have
The volume is, well, it's not conserved exactly, no.
You'll have small
But there's a big function, the Hamiltonian, which is like sort of like an energy. And it contains terms that you choose to put in there. Generally, there's a term that penalizes the area for straying too far away from some target area. So the cell on the whole will get a bit bigger and smaller and fluctuate. Yes, but by and large, it'll more or less stick around its target area.
And so in terms of what you put in, it's just through the chemical interactions of diffusion and reactions, and there are no mechanics between them.
Now what we did in particular, what we did in particular is that in each of these hexagonal sites, we solved all of these equations. Okay? So all of these equations were solved, and in particular, close to the edge, well, it was solved in the interior, but in the edge, ARP23 will get activated and will produce actin filaments, and they're going to be in all different orientations. And then whether or not that piece extends or retracts depended on some balance between the number of barbed ends that are correctly oriented, meaning pushing against that edge, so in a direction, let's say perpendicular to the edge, and the local amount of myosin, which is really, rho is the, this thing right here.
Yeah,
the sort of
the
represented
reactions
myosin.
are dependent on just these concentrations, and
Yes.
they are mechanically isolated from their neighbors.
Yeah,
Except from the diffusion.
right.
One question you can maybe you address, but cell polarization is crucially linked to growth.
In what I've discussed, I haven't really talked a lot about cell growth. I've talked more about cell motion, cell polarization, and motility, meaning how the cell gets a front and a back, and then the front assembles actin and starts moving, protruding outwards, and the back contracts and so forth. In cell growth, they may very well be implicated in that as well, but I haven't talked about that. Yeah. And I don't know about it very, very much.
What's the role of data assimilation in these types of
I'm sorry, say it again.
What is the role of data assimilation?
So far, all the data I've shown you were basically either for the cell wounding experiment in the beginning or the HeLa cells in the microfluidic devices. So, I mean, ideally, it would be nice in the future if we could actually figure out what sort of functions to put here that connect to what's actually known about this GEF and GAP proteins that activate and inactivate the GTPases. So that may emerge. It's very problematic because it's very hard to measure these things experimentally.
In some sense, could be one way of doing mathematical modeling.
Yeah, well,
I mean, by having large
sure.
amounts of data and then imagining the largest possible mathematical model that you have, and then trying to find out what are the important parts of the mathematical model by using the data.
Mm-hmm. Right.
It's
So
not
far,
not just parameter identification.
oh, for
For
sure.
example, this F that you showed, this graph of this F that you showed.
Yeah, all these functions are completely made up.
Right, exactly. So, but they don't have to be made up. They can be made up out of
data, absolutely. That would be
parameterized by some
fantastic.
parameters which are becoming
Yeah.
the objects of which. Yeah.
That would be fantastic. I mean, this is just a plausibility argument. I mean, we had no data, and so we made it up. We made up a story. But I think that there are certain features here that are likely to emerge. Oh, let me say one thing. For instance, we made certain assumptions that put all the interesting nonlinearity in the activation rates of the GTPases. But we showed that you could just as easily set these to be constant and put all the interesting stuff into the inactivation rates. So, I mean, we looked at variance, but for sure, if there was a way to get the correct functions from data, this would be way better.
Well, in the context of dicty, there is a lot of that known. So, for example, we had the GEFs and the GAPs included. I didn't go through the detailed model, but
Right.
they are in there. And the point is that you could constrain things quite a bit by reconstructing the time sequence of the response. So I showed you this biphasic kind of response.
Right.
Well, in one cell type, G alpha null, it turns out, that secondary response is completely obliterated. So that tells you a lot about what the underlying model has to do.
Yep, maybe dicty will be the first cell that gets completely mapped out like this. That's quite possible.
I have just a naive technical question, but your method of perturbation, how does it relate to the classical linear perturbation analysis if you consider just perturbation of
Right.
infinitely wavelengths?
Okay, that's a very good question, and I didn't have time to go into the details there. So, Turing analysis, for example, is a kind of linear stability analysis, and we commonly use that for reaction diffusion equations, right? The relationship is that Turing analysis can tell you what happens to very small noise, right? Whether or not small noise is going to grow into something interesting or decay. Turing analysis cannot do any justice, cannot handle perturbations that are larger than a certain amplitude. And to some extent also, Turing analysis is more challenging and doesn't scale up that well. So to do it for a pair of PDEs, that's not too difficult. You find eigenvalues of a certain, matrix, you see that, etc. When your system has 10 PDEs or more, it becomes a challenge, right? So the advantage here is, first, it gives you more insight into both the Turing regime and the threshold regime, because you're looking at pulses which are narrow but of arbitrary height. But also, you can hook into all the bifurcation software, because you're basically dealing with ODEs. So you can put it on whatever is your favorite bifurcation package.
But then you're going to miss potentially the modes which does not happen at infinite wave.
Right. It tells you nothing about the spatial behavior. It does not tell you about which modes would be unstable. It doesn't tell you anything about to expect, low or high spatial frequency. To see that, you've got to go to your PDs or your other favorite tool. The only thing it does, but this is quite a lot, is it gives you a feel for parameter manipulation. What range of behaviors am I expecting as I change a bifurcation parameter? Do I expect to see regimes of, threshold type, Turing regime, and so forth? Okay.
Perfect. Thanks.
Thank you.